My name is Michael Banks, and I work for Oregon State University, part of the Coastal Oregon Marine Experiment Station that is based out here at the Hatfield Marine Science Center in Newport. My academic home in Corvallis is in the Department of Fisheries and Wildlife, and then I also direct an institute called the Cooperative Institute for Marine Resources Studies. How did I get involved with Project CROOS? Well, my boss, Gil Sylvia, came and knocked on my door one day in 2005 and shared with me that Senator Wyden had visited with him and asked if the university could help with a problem associated with the Klamath River fall run and fishermen crisis. These stocks were doing really poorly, and it was apparent that the fishermen were about to be swept off the ocean completely, full stop. Of course, that would be very much a hardship for them. And we were basically developing a genetic baseline that enables one to ID a random fish caught in the ocean to their most likely origin, so the region of where they most likely spawned, from California all the way through Oregon, Washington, Canada, and up into Alaska. So I said, yes, Gil, I think we have some tools that might be able to help us with this problem. I think we need to use these tools to learn more about the distribution of fish in the ocean, and I think we could do a wonderful collaborative project with the fishermen here and get them back out on the ocean, maybe in a much more targeted way, having them focus on fish that we want them to catch and away from endangered stocks.
So was there something specific that really got you excited about the project? What came to you in your mind first that you wanted to be involved with it?
Well, to be quite honest, it's a dream I've had since 1994, when I first began to work with salmon. I was in California at that time, working with the endangered winter run Chinook salmon, and that's when we first developed these markers that we now are using up and down the coast. And I always thought that these tools that we were developing would be really, really informative for the fisheries. And it took about 10 years before I really had a wonderful opportunity to apply them in the ocean. And that was it. I guess if I think about Project CROOS, though, that what the project has become now, what really is very most exciting and amazing about that is the incredible sense of goodwill and hard work and cooperation that has resulted among people on this project from a very diverse numbers of different walks in life. They are fishermen, various types of scientists, various agency people. There's just so many different people really integrated and very interested in this project, all to learn about the behavior of fish in the ocean and how we might preserve some and go ahead and catch others that can withstand that. So this was just a bit of introductory comments. During the marine period, it's well known that Pacific salmon routinely migrate very long distances. Some even, say, from some of our stocks here in Oregon, migrate all the way up into Alaskan waters, passing Canada along the way. And so what happens is that they quite often are caught in areas of the ocean that are way beyond the jurisdiction of where they actually originated from. So actually most of our Oregon salmon are caught either in Canada or Alaska, and my colleagues up there love our Oregon salmon because they're very, very highly valued in their fishery. So that creates an issue with regard to their best management. If they getting caught in regions completely different from where they spawned, then it basically requires lots of very sophisticated cooperation amongst all the different governments and jurisdictions. So in consideration of this, and with the numbers of salmon declining, USA and Canada beginning around about the mid-80s, agreed to cooperate in the management of and research associated with the enhancement of Pacific salmon. And there's a body called the Chinook Technical Committee that has taken responsibility for providing data used to specifically address fishing quotas among the various parties and manage the system sustainably so that we have salmon into our future and our grandchildren can enjoy these fish too. A major tool that has been utilized by this body I've just described to get an idea of how the salmon are migrating and where they're occurring in the ocean is a tool we call coded wire tags. So coded wire tags are a very neat tool that has been used by the management bodies for observing where salmon are in the ocean, in that these are little pieces of metal that are placed in the heads of juvenile fish as they leave the hatchery, with little marks on them that enable you to identify exactly where and when those salmon were released. So it's actually very precise information that you can get from those fish, and that when these fish are returned to the ports after being caught by the fishermen, they are scanned for presence of these tags, and those that have these tags, their heads are chopped off and sent to various labs that exist up and down the coast to read them. And that's basically how we have learnt about the distribution of salmon up until now. This is a wonderful tool and remains very powerful because of the precise information you get from this information, from reading these tags. But there are issues with coded wire tags, and one of the major issues is sampling. It's not feasible to put little bits of metal into all the fish that are out there in the ocean. And more precisely, we don't have access, we don't handle wild fish, and increasingly we've gotten really interested in measures to preserve wild fish and learn more about where wild fish are in the ocean. So that's why we now have developed this genetic tool and this process called genetic stock identification, because all fish have genetic tags. It's just something that they have as part of existence, and it enables us to actually pick up any fish from the ocean and make estimations as to where it most likely came from. So that provides just a whole new different potential for learning different things about the ecology of salmon whilst they're in the ocean life phase. I've spoken about this tool as a genetic marker, and I guess I wanted to get into more specifics about that. We utilize a specific region in the genome called microsatellites. And by the word genome, I mean basically the blueprint that exists in each cell to code for all its basic characteristics. So microsatellites are within that blueprint, and these are really short little fragments of DNA that are unique in that they are characterized by a repeat of a specific motif of sequence that is repeated. DNA has only 4 basic building blocks. We call them bases. And these repeats may be, a CAG, repeated a number of times. Or ATC is another microsatellite. And these microsatellites, because of these repeats, when DNA is replicated during cell division, the enzymes that replicate DNA sometimes make a mistake. They might add an additional CAG, or they might drop a CAG. And when that occurs, there's a change in the length of this short fragment, which is the character that we assay when we assay microsatellites. In genetic terms, we speak about these regions as being regions of high mutation. So you can think about them as kind of like packs of cards, and I've got some cards here to show you some of the idea. So here's 2 different packs of cards. Let's imagine they're the same pack in 2 different populations. And I've spoken about these different lengths, which you can think of as being these different cards. And because of what occurs over time, and because different mothers just happen to be maybe more fit than other mothers, they might choose to be better at choosing a buck that's more fit than other females, or they might just be lucky in that they've given their eggs a really special place in the nest, and those females do very well. In certain populations, you might get a real high frequency of some of these cards. So in this unusual pack that I've created for this demonstration here, we have a whole lot of tens of diamonds. So this might be fish in the Rogue River. So when we assay this particular microsatellite and we find a whole lot of tens of diamonds, we have a pretty good idea that those are most likely Rogue River salmon. Whereas fish from the Klamath might not even have any tens of diamonds at all. And so when we don't find any tens of diamonds, we could maybe estimate that those are most likely Klamath fish. Of course, this is just one locus, and we don't have much power just with one. The word locus slipped in there. That's the specific region in the genome. And because of us wanting to have confidence in our results, we actually assay a number of these different microsatellites, and those give us greater power for the discrimination. So I wanted to talk a bit about actually the early days when we first isolated these microsatellites in Chinook salmon. I was a researcher in California at that time, just north of San Francisco Bay, at a laboratory called the Bodega Marine Laboratory. I was in the laboratory headed by Professor Dr. Dennis Hedgecock. He used to call his lab the Intergalactic Marigenetic Center. And that's where we isolated the first Chinook microsatellites. We were in a project there trying to find ways of identifying the endangered winter run Chinook. We were actually responsible for a captive broodstock of this endangered winter because their numbers had really decreased. This was in 1994, at a time soon after the Sacramento River winter run Chinook was listed as endangered. Numbers of returning winter run Chinook had gone down to really disturbing numbers. I think they were in 91, there were 191 parents returning. In 94, there were 184 parents returning. So we actually had a permit to take care of, I think, about 30 of those returns as a captive broodstock, trying to create conditions to breed them favoring genetic variability and protect the survival of those juveniles, so that should there be a complete failure in the wild, we could restock this specific life history back into the system. So our task was to try and find means of discriminating molecularly this winter run Chinook from the three other runs that exist in the California system. They also have fall run and late fall and spring run, a total of 4. The runs are named after the peaks in the time when the major numbers of fish return to spawn. And as I just detailed, the peak of winter was in some years only 190 fish. In those early days, and as we started to extract these microsatellites I've told you about from the genome in these different runs, the very second microsatellite we got our hands on, one that we called OTS2, Oncorhynchus chao-chai 2, we found one that had a fragment that was 66 base pairs in length, highly frequent in winter run. In fact, 80% of the winter run fish we looked at had this particular little fragment. None or very few of fall run, late fall or spring run had this little fragment. The frequency in winter run was 80%. In all the other runs it was less than 5%. So actually all our genes exist in 2 copies. We get one gene from our mom and one gene from our father. But given that 80% frequency in winter run, the frequency of which we might expect what's called a homozygote, having a 66 base pair from your mom and from your dad, is the product of that frequency. So 0.8 times 0.8 is 0.64. Still greater than 50% of the individuals would be having the 2 copies of that gene within their genome. Recall I said all the other runs had a frequency of less than 5%. Let's just say it's 5%. That's 0.05 times 0.05. So the frequency of finding a double copy in, say, fall, late fall, or spring is 0.05 times 0.05 is 0.0025, which is 0.25%. That's 1 fish in 400. So you can see when we found a homozygote 66, we're pretty likely that's going to be a winter run. And just to give you a bit of an idea of how the technique works. And we do that at multiple microsatellites, so we compound the power that we have.
So can you give us a little bit more on this, the scientific excitement of, like what it was? I mean, that's a great scientific adventure.
Well, it was actually a very interesting time in my career, because as we discovered that, I mentioned we were in charge of this captive broodstock. And as we began to learn that this OTS2 was highly diagnostic for winter run, we found evidence that some of the fish that we were provided for the captive broodstock, according to the genetics, didn't look to us like bona fide winter run. So actually, we had been provided some spring run amongst that early year of captive broodstock, which of course was a very controversial moment, because we were now dealing with this endangered species, and now we had hybridization, and the work was all very new and unproven. And so we just said, well, this is what the science tells us. Let's keep going and see whether other markers continue to affirm this finding. And indeed we did. As we characterized other microsatellites, we were able to affirm our suspicion, and we actually had to purify that broodstock so as to not hybridize amongst this endangered species. So that was, in the science process, we rely on our peers, and the work was something that was brand new that we discovered, and so we had to get it peer reviewed, and that takes time. And we had to let the hatchery managers know that there was this issue at hand, and it was an issue of controversy for a while, but something that we communicated about and survived. It was an interesting moment. Anyway, so those were the start days. Let's go back to 2005, now 11 years after those early days, where I told my boss that we had this GAPS baseline.
What's GAPS?
The Genetic Analysis of Pacific Salmonids. It was a baseline which colleagues from California, Oregon, Washington, Canada, and Alaska collectively created, characterizing fish from each of our regions so that we could ID fish back to their most likely source. We obviously needed to work together so that our baseline was standardized. And this was quite an interesting exercise too that I'd like to talk about in a little while. We developed this standardized DNA baseline, which we offered as a tool to be used alongside the coded wire tag tool for learning about fish in the ocean. There were actually 12 genetic laboratories involved: Canada, Oregon, Washington, Alaska, various states in the US, Idaho also. And we chose 13 different microsatellites as the base data. Those 13 microsatellites have of order of about 500 different alleles, more than 40,000 salmon assayed across that large region so that we could know the specific signature of each of the regions. And more than 270 populations are now in our baseline, so quite a tremendous resource. There were some technical issues and standardization things that I'd like to talk about that were kind of interesting. we all use this technique called the polymerase chain reaction, which won a California surfer a Nobel Prize. He's the one that invented this technique. And actually my professor, Dennis Hedgecock, was one of the early professors to learn this technique. He went to sabbatical in the company where this technique was developed. The polymerase chain reaction is a method by which DNA is double-stranded, and basically through going through different cycles of different temperatures, we denature DNA into single strands, which sort of releases the code, and then we, through specific primers for the genes we're interested in, the microsatellites we're interested in, we amplify across those codes. And we repeat that cycle multiple times until we have billion-fold copies of the genes we're interested in. Now, that's quite a sophisticated technique, and you can imagine that 7 different laboratories from California up into Alaska may have some different means by which they achieve that complicated technique. And so we had to talk about all our different techniques and decide which ones we would use in common, and that was a bit of a task. So choosing the specific procedure to be used, because of course we all have our favorites, and genetics is a pretty high fidelity technique, and we each are our own specialists, and we each have our own pets, so we had to sort that out. And then we discovered also that we each had different microsatellites that we preferred to use. So we had to decide amongst all this lab of probably more than 120 different microsatellites, which were the best 13. And of course we all had different opinions about that. So yeah, what you might ask, so what is a good microsatellite? Obviously there are different parameters of interest here. One is how easy is it to amplify and score. Obviously that's something that needs to happen. The second major thing is how many different characters does it most commonly reveal among the different populations? And are those characters different in different populations? Although there may be many different characters, if they're not very variable among the populations, then it's not much use. So unfortunately, these parameters are kind of quite often in opposition with each other. Markers that are very easy to amplify might not have many different characters. Markers that have many characters and are highly informative across populations might be very difficult to amplify. And so that was and of course different labs had mastered different microsatellites, and so actually deciding on these 13 was quite a task. And as I was just describing, because I was involved in the early isolation of these microsatellites, I was one of the investigators that I guess had experience with a lot of them, and I did pretty well in that. Actually 7 of the 13 in the baseline are out of the lab I used to be in, and out of what we're developing here. And it was kind of interesting for me because amongst those 7 there were 2 that were not my favorites. Although they came out of my lab, they weren't nearly as informative. I knew that these markers were good and they were easy to amplify, but I knew that there were others that had been developed more recently from other labs, actually, that were more informative. And I strongly said, although I'm shooting myself in the foot, we need to get rid of those 2 Banks markers and replace them with something else.
10 of diamonds.
Yeah. But because many of the other labs really liked them and they were easy to amplify, I wasn't able to succeed in shooting myself in the foot. So that's how it is now today.
Could you explain why they might like them in addition to being easy to amplify?
Well, it's because we all, I guess, see the world from our own particular windows. So here I am in Oregon, and I have also experience in California, so I'm very interested in markers that do well in Oregon and California. Someone in Alaska is more familiar with how markers are in Alaska, and so I guess that's why we have different opinions. And I think someone said, well, Michael, even although there are only 2 alleles in OTS3 in California, so it doesn't tell you anything about California fish, there are 15 up here in Canada or Alaska. And so we like that. And you had to, it was, really it was an interesting experience of reaching compromise amongst all these different parties to come up with a collective tool that would allow most valuable broad spectrum ID of salmon across that range. So obviously I'm sure many of the other players could tell you similar stories about how they had to accept maybe not their most favored choice as well.
How long did it take to decide on the 13?
Start it as if you don't hear her say the
I would guess that it probably took us about 1.5 years to 2 to actually go through that decision process. There's actually a paper written by Paul Moran out on that, and maybe some others as well. Paul Moran is a scientist in Seattle who's taken a leadership role in the development of the GAPS baseline. And there are many players.
Can you approach the challenge of the blind tests?
Yeah, I guess the next and last one of the many technical issues I wanted to talk about is once we had decided on those 13 best microsatellites, we also had to ensure that we were scoring those 13 microsatellites correctly in California, Oregon, Washington, Canada, and Alaska. And we all have our own really high fidelity machines to score them. And we found that although these machines may cost $300,000 or $400,000, they don't necessarily always give you exactly the same marker, I guess the same call for the same marker. So we had to do a process we call calibration, where we make sure that a particular genotype scored in California is scored in the same way as the genotype scored in Alaska and Canada and Washington, Oregon. And that's what we call the standardization. So what we utilized to check ourselves on that was what we call a blind test, where we had a tray of 95 fish that somebody else prepared and then provided to us, and we all scored them in each of our independent labs, and then we collectively submitted our results to a different party who saw whether we scored all the same, which you can imagine is a scary thing for a group of high fidelity labs to do, because you never know whether you're the one that agrees with more of the others than one that disagrees with more of the others, and who really knows which one is right. And so, of course, majority rules, and there were some labs that agreed with each other more than others, and so that was a tricky thing to manage. So I've been through some of the technical issues. I now want to just say that it's been a well worth exercise because of the immense power of the baseline. We now have determined unique genetic signatures of the salmon populations up and down the coast. We've established a broad spectrum standardized baseline across the Pacific Northwest, which is just fantastic for its enabling power. We can now have fishermen go and catch fish anywhere in our Pacific Northwest, have them deliver those to us as soon as they can, and within 24 hours, we can tell them where we think those fish came from, which is pretty fantastic. I didn't mention that the coded wire tags, as you might imagine, the fish have to be caught, they have to be identified in the port, the heads have to be chopped off, they then have to be sent to the lab, and it's not possible to read those heads very efficiently. So, I mean, there are many labs and many wonderful people that do that, but typically the turnaround time on synthesizing that sort of data is about 6 months. Way different to what I just said, 2 days that we can do with the genetics. So that's just really fantastic. And just to give you an idea of what we can now do, we can most often match that individual salmon to its most likely region of origin. So it might be a region in Oregon, or a region in northern Oregon, or central Oregon, or southern Oregon. And in some cases we have enough power to identify fish back to their river. The Klamath River is an example of a river that's rather unique, and we actually have a greater than 95% chance, that's how much we trust the data, for a Klamath River identification. Further than that, within specific rivers, we sometimes have actually enough power to identify specific life histories within that river, and the winter run from the Sacramento River system is an example of that. They are so unique and different genetically that we have very strong power of identifying them with strong certainty. So when a winter run comes up, we can not only identify it to a region in California, but that is a winter run fish, which is rather useful given that that's one of the endangered species. So it really helps with our management of them.
Who are the consumers of this information?
Lots, I guess. The Pacific Northwest are the consumers of this information, because we are utilizing these tools to help with management, help with allow fishermen access to stocks that they previously might not have had access. We're even making the information available to marketing. So it's actually possible for you now to go into Portland and purchase a salmon that might have a tag on it, which you can go and utilize to consult a web page on your home computer, and that would likely tell you where your salmon was caught and who the fisherman was, and how, what his history or her history might be, and that's actually pretty tremendous, very exciting. So I would say the consumers are the people of the Pacific Northwest.
One of the most difficult parts of your project is determining the fish that are under the boat. The fisherman's waiting to decide, to get the information as to whether or not he should go ahead and fish. And how can you tell, how do you approach the problem of, are these all Klamath fish, or is there a mixture of Klamath, Rogue, Umpqua, or how many samples do you have to take, or what's
Sure.
the approach to do this?
Well, that's the essential million-dollar issue we're trying to learn about, really the distribution of the different stocks during the ocean migration. And really, that's affected by a number of different things which actually are changing. So the ocean is a very dynamic environment, and also time is at hand in that fish likely behave in different ways at different times of the ocean life phase. And when they're at the stage when they may be deciding to go back to the rivers to spawn, they likely, because they have to go back to specific rivers, they likely order their distribution a lot more than at other times. And we are basically trying to do enough research to get a better understanding of the connection between environmental conditions, time of year, and these different stocks as to how they may vary in terms of the distribution. We have some tantalizing information from some of our observations that they may have very specific run or river-specific aggregations at certain times, and they may be much more mixed at other times. And that's a really tricky thing for us to quantify, and so that's basically why we estimated that we probably needed a 5 or more year collection of data, which would likely span different Niños, La Niña environmental conditions, and likely give us information about how fish may behave under different conditions. And so, I mean, there are cases where fishermen will say they've caught these fish at the same location, and when we look at them, they came from the same region. And there are other cases where they may have come from various regions. So
it's pretty exciting modeling.
Yeah, and it's really been an interesting lesson in terms of scope and size of the project we needed to entertain. It of course first began in Oregon. We had this tantalizing preliminary information, and then as we got more information we realized, hey, this is actually a much bigger thing, and it would be really neat to know about the distribution of these stocks on a larger scale. So that's why we encouraged California and Washington to join us, and now we do have, for the first time this year, we have very exciting data simultaneously from California all the way up north, and we're hoping that we can get a number of years like that, and with that kind of range, maybe we can get our hands on some of those questions that you're addressing.
Are you doing modeling not only of where the specific genotypes are in the ocean, but are you using temperature and all of the other kinds of data that are being collected?
Well, that's what one of the really neat things about this project. It is multidisciplinary. There are oceanographers, microchemists, geneticists, physiologists, all different people. Physiologists, all kinds of people are involved in that, and we're trying to synthesize across all those disciplines. So we would like to see if there's connections between particular environmental oceanographic conditions and where specific runs or genotypes might be. And really what's very exciting for me in this is it's an opportunity to learn how maybe the genome, different parts of the genome are involved with different things. One of the exciting things for me is a project that a Ph.D. student I have by the name of Renee, looking at how fish know where they are in the ocean, how they determine their orientation. And another exciting part of this project for me is that I see lots of application for research of another colleague of mine, Kathleen O'Malley, who studies clock genes here, which allow us to learn about the genetic aspects of the timing of some of these events. So when fish may return to spawn, or how they make decisions of when to finish their ocean migration, and that's their feeding migration, and return to their spawning migration. All these different events are controlled in space and time. In Renee's work, we after the space aspect, and in Kathleen's work, she's after the timing aspect.